Hello everyone, welcome to the online session. In this session, we are going to discuss about the molecular docking using various tools, AutoDock or GemDock or any online servers. I'm going to share screen now. So let me discuss about this protein preparation first, then later we'll discuss about the molecular docking in detail. Okay. So for molecular docking, we need two raw input data, that is proteins or ligand molecules. Ligand, it can be a drug molecule, it can be a peptide, and it can be a metal ions, or it can be any that DNA molecule also. Because the molecular docking can be done between a protein molecules or between protein and DNA, or protein and a drug, or DNA and a drug. It's like studying of the molecular interactions between the molecules. So that's called the molecular docking. So for that, here I'm going to talk about the protein or receptor ligand docking. So we need a protein file or a ligand file to do docking with this any small molecule. First, let me download the protein structure or a 3D file, 3D three-dimensional structure of the protein molecule from the protein data bank. So here I'm going to use one protein structure, that means that is 6Y84. 6584, I'm going to, this is a PDB ID of a protein. So when I click it, you can see there's a SARS coronavirus main protease enzyme, which is responsible for the binding of this virus into that human host cell. So main proteases, I'm going to download it, and even as spike proteins also there. So main protein is one of the targetable proteins in antiviral drug research. I'm going to use this main proteases now for this molecular docking studies. So click on this ID here. So when you click on that ID, you can see here the detailed information of this protease protein of SARS coronavirus. There are already ligand molecules are present here, some of the dimethyl sulfoxide and all those things. So that should be eliminated for docking research, because we want to use a different ligand, and if already ligands are present in the structure, that will create some problems. So we need to eliminate those ligand molecules from the structure. Okay. So how to download this protein file from this? You can go to the download files here. You can see here. Here download PDB format. Why PDB format? Because most of the tools in computational studies uses the PDB file format to analyze the three-dimensional structures. Okay, so PDB file format is a universal for computational studies or analysis. So I'm going to download this file, SARS coronavirus main protease in PDB file format. Okay. So this file will be saved into one particular folder. For example, here I just saved it in this file, this folder. I tell you what is gemdoc later. So now protein file is ready now. So I'm going to do now. I'm going to retrieve now ligand file. Ligand I'm going to use here the hydroxychloroquine. So one of the anti-malarial drugs. You can download this drug structure from PubChem compound database. We all know this NCPI PubChem database. It's one of the major database for macromolecule as well as small molecules. So PubChem databases, we can get the hydroxychloroquine in here. So hydroxychloroquine, you can type it here in the search column and choose here in all databases, choose PubChem compound database and search it. When you search, you will be able to see that list of this hydroxychloroquine in here. Hits, there are 13 hits are given here. So first one is hydroxychloroquine, and all others will be derivatives. So derivatives of hydroxychloroquine. So I'm going to download this hydroxychloroquine. So how to prepare the structure for molecular docking study? That is one of the important steps in molecular docking, because in the database we don't get a direct PDB 3D dimensional structure. So we may get 3D structure, but we'll get only in the different file format. Like you can see here, there are 3D formats are given here, 3D structure. But when you go to the download option, you will be able to see the only SDF and other formats. So we cannot find any PDB format here. So, but molecular docking, many software access only the PDB file format. So how to convert this file into PDB file format? That is one of the important steps here. So I'm going to use canonical smileys. If in the same page, if you scroll down, you will be able to see the canonical smileys. I just copy this canonical smileys and go back to this online server. That is, we say it is Corina. You can see the Corina online server, Corina online fast 3D structure generation server. Here I'm going to paste this in this column the smileys which we copied from the Perfume compound database. This is smileys of hydroxychloroquine. Smileys is nothing but already I mentioned the simplified molecular input lineage specification in previous online session. And this mileage can be used for many analysis, further analysis of 3D conversion or property analysis and the physiochemical parameter analysis of the drug molecules. Either you can use that 3D structure or you can use this simplified format of that structure. Okay, I just pasted here and just submit it. When you submit it, this tool will convert this hydroxychloroquine structure into three-dimensional structure. You can see here three-dimensional structure, and also you can download this file as a PDP file format and MOL file format. So we need a PDP file format in this case of docking studies. So it is downloaded here. I'm going to copy this file and paste it into the folder, particular folder which I created here. Okay. So immediately I just label it. If I'm using here one molecule, it's fine. So, but in case if you're using many molecules, you will be confused with the codes generated by the Korean online server. So immediately you need to label it. I attach a chloroquinone. Now we have two files here. This is a receptor file and the ligand file here. These two file how to do your docking. Okay, so we have a tool called GemDoc. So if online it is freely available, one of the easiest tools and will get good accuracy results, that is GemDoc. You can download it, Windows XP and different operating system it is given here. And you can download the file, you can easily download. It is not installable, it's standalone program, but still you can use it without installing the server. You can just copy paste. In that folder, GemDoc folder, I copy pasted um, protein file, a receptor, or ligand file, both the things. Now let us start the docking molecular interaction studies. So if you open the bin folder here, you can see that application file here, iGemDoc. Click on the application file here. Yeah. So you can see here the main page of this graphical user interface of this iGemDoc. There are two main options here: protein ligand docking screening or a docked post or post screening analysis. After docking, we need to do some more analysis, that is called the post docking analysis, to analyze the binding free energy, the molecular binding free energies, and the binding interactions and the binding patterns and binding mode and orientation of the molecule and the active site packet of the protein. All these informations you can get it after the docking in the post screening analysis. Now let us start with this protein ligand docking screening first. Okay. So in this you can see here there are three major options they given: a prepare binding site and a prepare compounds and a set path. Okay. Then start docking. So I'm going to upload here the protein file in this region. You can see the prepare binding site. Yeah. So prepare binding site. You can here you can upload this file, protein file. So when you click on that browse file, you can upload this file. This already we downloaded from protein data bank and prepared. That's okay. You can see the protein file is uploaded here. The coronavirus main protease file is uploaded here. And I'm going to upload now the ligand. Here, two things I have to tell you here. One is ligands and folders, two options are given here. For example, if you use a one ligand, you can use the ligand file here. You can upload it. Yeah, one ligand is uploaded. In case if you have multiple ligands, like more than hundreds or thousands of ligands, you can put all the ligands into one folder. That folder also can be imported here directly. So this iGEM dock, it's for multi-purpose. Here you can do protein ligand docking, or also you can do the virtual screening or isotope screening of the molecules. So you can do the protein single docking, and also you can do the protein multiple ligand docking. Okay. Here I uploaded one protein here, import it. Yeah. Yeah. Okay. Okay. You can see that I uploaded protein here. I uploaded a ligand here, and also path output. It will generate some of the results from this analysis. That result should be saved in some place. Otherwise, it may saved somewhere, then very difficult to track it, where that file and how to analyze the molecules. So you need to fix a path properly. So here the download the same folder, I'm going to use it, gemdoc file, gemdoc output folder. If you click that gemdoc output folder, in that folder it will be saved. So coming to this parameter, genetic algorithm GA parameters here, you can the population rate, generation, number of solutions. If you want to do the standard docking, you can do. Still, it is stable docking. You can see the increase in these values, the population size generation. That means it will check the more conformations and orientation of the molecule in the active site of the particular protein. And also accurate docking is also there, and the drug screening also there. Yeah, you can also quick docking is a rough docking, or you can custom all those things. Custom dock. You can just reduce the number of sites. For example, population size here. I just go for this quick docking now, and here I just put only the 30. Uh, generation and one and population size is 20. Start docking. When you click on the start docking, you can see that here the calculations really happen here. It will just check the conformation of the molecules and their binding mode or orientation of the molecule and the active site of the packet. So here I put 30th generation. Now the current generation is 3 now. It is running now. So maximum this calculation is run by the genetic algorithm, as we mentioned here, the genetic algorithm parameters. Okay, this is one of the major docking algorithms. So many of the tools are used this developed based on this genetic algorithm parameters. In fact, in Autodoc also the developers use the genetic algorithms. They develop the genetic algorithms to calculate the binding free energy and to analyze the molecules and their binding interactions. Okay. So, and a few more options they're given here, set scoring functions and other things. For example, if you're working on one of the important feature and interesting thing in the GemDoc is, you can see here when suppose if a current cut, electric cut, somewhere the docking is disturbed, the docking might be stopped. So in such case, you can use the view latest. If you want to continue the docking from the same places, the position where it is stopped, you can use the view latest process, and also after that you can resume process. So it will start from the place where it is stopped. Okay. So this is the interesting thing that very helpful things, very helpful parameters, features they given here in this GemDoc. Okay, there are many other features. It's so easy. If you use many other tools, there you have to follow many steps to prepare the molecules to do the docking. But when you use the GemDoc, you can see only 3 steps you have to follow very carefully. That is uploading the receptor file and uploading the ligand file here, and you can once uploading you can see the number of molecules one year that it will reach and the number of selected compounds 1 year. The current generation is 20th generation is going on. Yeah, so all this information, if you provide properly, the tool will calculate and run. If you want to check one or two molecules, then you can use that slow accurate docking so that it will check properly. You can do that. And one more features here, I've just put it for blind docking. In case if you want to do active site docking, you need to provide the active site information in this prepare binding site user interface. That interface, if you provide that active site information, then the tool will check the interactions with that only active site amino acid residues and give us the results. Okay. So 30 generations I have given here. So 30 generation is done now. It's going to be done now. You can see the 29 and also number of solutions one only I given here. So now the docking is going to be completed, finished. So once it is done, you can see the docking process is completed. You can see the docking process finished now. Click OK. Now docking is done now, and we are going for now the post-screening analysis. After docking, we have to do some of the analysis that is called the post-docking analysis. That is, here they given view docked post. Click on this button, so it will generate the binding energy. Yeah, all these details we can see here: binding energy, van der Waals energy, hydrogen bond energy, electrostatic energy. And still, if you want the docked post, if you select and see the interaction analysis, what are the amino acids are interacted with this hydroxychloroquine? The active site of this particular coronavirus protease protein, amino acids, active site amino acid residues interact with the hydroxychloroquine. So this information we can get. Still, if you want to see that their interactions, click on this view option. Yeah, view option. There you can see that exactly how the molecules, where exactly it's bound, and their interactions details. Okay. So still you can just visualize and see that those amino acids interacting with the hydroxychloroquine. Yeah. This image also you can also save into your local drive. That means you can see that energy, and if you want to save, you can just save it in text format or any other Excel format also is possible. Okay. So and the clusters also, there's clusters. If you use the multiple ligands, then you can just find out clusters. Okay. So these are the few things which will be very interestingly noted. Yeah. So these are the major steps. So in this session, we have discussed 4 major things. One is how to download a ligand structure, how to prepare a ligand structure. Here we have used cytoxychloroquine, and how to download a major molecule, macromolecules like protein structure from protein data bank, and how to prepare it. And after that, how to download the exam doc, how to do the docking analysis. Third one, from the docking analysis, what are the parameters we have discussed that we have seen. Then after docking, the post-docking analysis, they're finding out that energy, binding free energy, and their interactions and interacting amino acids, all those things. These four things should be there in the molecular docking studies and the molecular interaction studies. Yeah. So with this, I just wind up this session. I hope this session will be useful to you. Thank you.